guys, it's me, Chris. Today we're going to talk about lipid metabolism, specifically triglyceride catabolism or triglyceride breakdown. We're going to go over a series of videos. Specifically, we're going to talk about the mobilization of fat in adipocytes or fat cells. Then we're going to talk about glycerol metabolism, fatty acid activation, the transport of these fatty acids into the mitochondria, and then the last thing we're going to talk about is beta oxidation. So, as you know, we have fat in our bodies as energy stores. And as you know, fat is hydrophobic, so it can compact, and it has a high energy yield of 9 kilocalories per gram. But how do we utilize this energy when we're starving or stressed or fasting, things like that? Well, in our adipocytes, we have these triacylglycerides. And in our bodies, we need to break this down in order to get energy. And to get this energy, we need to break this triacylglyceride down into biomolecules that can enter glycolysis or the Krebs cycle. So keep that in mind. Throughout these series of mini videos, I'm going to make this a flow chart so you can see how all these processes are connected. Okay, so in this first video, we're going to talk about the mobilization of fats in adipocytes. Anytime you hear adipocytes, it's just a fat cell. So we have triacylglycerides stored in our fat cells. With the addition of water and something called hormone-sensitive lipase, triacylglycerides are broken down into glycerol and fatty acids. Now we're just going to focus on hormone-sensitive lipase because it's the most important part of this step. Hormone-sensitive lipase is an enzyme that breaks down triacylglycerides into glycerol and fatty acids in response to a hormone, hence the name hormone-sensitive lipase. It's actually part of a cascade. This cascade, the hormone, which can be epinephrine or glucagon, stimulates adenylyl cyclase with the G protein, and this produces CAMP. The production of CAMP activates protein kinase A, which activates hormone-sensitive lipase and perilipin by phosphorylation. This phosphorylated perilipin allows hormone-sensitive lipase to access the surface of the lipid droplet, where it hydrolyzes triacylglycerides into glycerol and fatty acids. Then fatty acids leave the adipocyte, bind to serum albumin, and carries the fatty acid to the bloodstream. So that's the cascade. It's a good idea to know how hormone-sensitive lipase works, but overall in this step, the mobilization of fats in adipocytes, the best thing to know is that triacylglycerides with the additional water are broken down into glycerol and fatty acid by hormone-sensitive lipase. Okay, so in the previous video we saw that hormone-sensitive lipase breaks down triacylglycerides into glycerol and fatty acids. There's a lot of processes involved with converting fatty acids into usable energy, so right now we're going to just stick to glycerol metabolism. So right now you have your glycerol from the previous step, and the first thing you're going to do is you're going to use glycerol kinase. And hopefully a workable marker. There we go. And remember, since it's a kinase, it adds a phosphate group to the glycerol. In order to do that, you need energy, so we're also going to use ATP. And this gets converted into glycerol 3-phosphate. I'm just going to... I'm just going to put the abbreviation for it. So once we have this, the next thing we're going to do is use glycerol phosphate dehydrogenase. And when we have dehydrogenase, we know we use NAD and NADH. And right here we get dihydroxyacetone phosphate. Now this should look familiar to you because it is in glycolysis, but you can't go through with glycolysis until you use triose phosphate isomerase and turn this into glyceraldehyde 3-phosphate. Then it can go through glycolysis and glycerol can then be used as energy. But this doesn't count for a lot of our energy. It only counts for 5% of energy from triacylglycerides. Now we're going to talk about fatty acid activation and see how fatty acids are used as energy. The main source of energy comes from these fatty acids. It counts for 95% of the energy from triacylglycerides. The main tissue that use these fatty acids for energy are the liver, heart, and resting skeletal muscle, not the brain. That's a question on the test, so please keep that in mind. So after leaving the fat cell, the albumin carries these fatty acids into the bloodstream, and the fatty acids enter the myocyte, or just a muscle cell, by a transporter. So now we're in the cytosol of the cell. In order to break down fatty acids, you must activate them, and it takes only a couple of steps. So you have this fatty acid, and if you use fatty acyl-CoA synthetase, and remember, when you have synthetase, you need energy, so you're also going to have ATP in this step. And this turns into fatty acyl adenylate. Before I continue, there's a key thing to know about synthetase. You must remember it's a thiokinase. Thio means sulfur, and kinase is anything that adds a phosphate group to something. That's a good thing to know for the exam. Dr. Cohen really likes sulfur for some reason, so he can ask questions on that. Alright, so once you have your fatty acyl adenylate, you're going to use fatty acyl-CoA synthetase again. But instead of using ATP this time, you're going to add CoAsh. And the AMP from this step is going to come out at this step. And your final end product is going to be fatty acyl-CoA. Alright, so this is your end product of fatty acid activation. Fatty acyl-CoA is in, this end product is in the cytosol of the cell, and you need it to go into the mitochondria for oxidative metabolism to occur, and it can't enter the cell like this. So in our next video, we're going to be talking about the transport of fatty acids into the mitochondria. Okay, so we finished fatty acid activation. We have fatty acyl-CoA left, but this is not suitable to transport through the mitochondrial membrane. So we need to change it so it can transport and get it into the matrix so we can further metabolism. So we have fatty acyl-CoA, and the two things that you need to know for this step is that you need carnitine and carnitine acyltransferase. This is my little diagram I showed. You don't need to know the details of this, but I kind of think it's a good idea to have a background of how this happens, just so you can understand it better, instead of just saying, oh, transport happens with carnitine and carnitine acyl transferase. At least you'll have an understanding. So pretend this is like a little cut of a mitochondria. You have your outer membrane here, your inner membrane and the matrix, and right now we're in the cytosol and want to get to the matrix. So you have your fatty acyl-CoA, and with carnitine, so and carnitine acyl transferase, the one on the outer membrane is 1. You don't need to know that, but it's a good idea to have an understanding. We're going to get fatty acyl carnitine. And this is actually suitable to transport through the membrane. So this is going to go through the membrane, through the transporter, and before it actually even goes through the transporter, it's going to go through carnitine acyl transferase 2 and turn this back into carnitine. If I could spell, it would help. And fatty acyl CoA. Now carnitine can get transported back to the cytosol to keep this going, but what you really want to know is that you use carnitine and carnitine acyl transferase to get fatty acyl CoA through the membrane, so you get fatty acyl CoA into the matrix of the mitochondria. Okay, so we've transferred the fatty acids from the cytosol into the mitochondria. So now what we're going to do is beta-oxidation to further metabolism. Beta-oxidation takes fatty acyl-CoA and removes 2 carbons from the carbonyl end of the fatty acid to give acetyl-CoA. That should sound familiar to you, but we're not going to get to it just yet. The reactions in beta-oxidation are centered around the beta carbon, hence the name beta-oxidation. And it's easier for me to explain all this to you if I actually use a fatty acyl-CoA. So I'm going to use palmitoyl-CoA, which is a fatty acid with 16 carbons. So I didn't actually write out the whole fatty acid chain. It would continue on until you get 16 carbons in the fatty acid. For a couple things I want to point out: which one's the alpha carbon, which one's the beta carbon. You're going to start from your carbonyl. So this is your alpha, and this is your beta, and we're going to pay attention to these 2 carbons. So there's 4 steps in beta oxidation. The first step, we are going to use acyl-CoA dehydrogenase. And when we use this dehydrogenase, we're going to use FAD, which will then become FADH2. And our end product will look something like this. Okay, so obviously something happened. Basically we had an oxidation that made a double bond between the alpha and beta carbons. So what I want you to look at is right here. And when I say oxidation, oxidation is either when you add oxygens or remove hydrogens. As you can see, we started off with 2 hydrogens on each beta and alpha carbon. Now we only have 1 on each in a double bond form. So what Cohen really wants you to know is that an oxidation happens in the first step between the alpha and beta carbon. By the way, you don't actually need to know all these you don't need to know like this is what happened, this is what happens. I just feel like it's easier for you to understand that, a double bond happens here and that's why it's an oxidation. It just helps you understand it, but this is a little bit more detailed than what you really need to know. So for the second step, we are going to use enyl-CoA hydrates. And since it's a hydrate, we're also going to use the addition of water. And our end product looks something like this. So basically in this step, it's a hydration. That's what Cohen wants you to know. You added water. Specifically to your beta carbon. Why does not look right? Oh, oops. There we go. To your beta carbon, you added OH of the water, and then to your alpha carbon, you added another H. So that's just telling you where it went. So second step, no, it's a hydration. Third step, I'm running out of room, so I'm going to erase this, guys, so sorry. Okay, so in our third step, we're going to use beta-hydroxyacyl-CoA dehydrogenase. And in this case, we're going to use NAD, which will then form NADH and another hydrogen. And our end product looks something like this. So remember how we had the OH and the H here, and then the H here? Well, basically what we just did, we just oxidized it. We removed the hydrogens. One of the hydrogens went here, and the other one went here. And we turned the beta carbon into a ketone. So that's what he wants you to know. He wants you to know that another oxidation happened, but I would say it's really important to know that this one becomes a ketone. I think that was an exam question one time. So I'm going to put oxidation. In parentheses, just know that the beta carbon became a ketone. It's actually a good idea to remember what you're starting from and what you're ending from and what you use. I would keep that in mind with the beta oxidation. So that was step 3. We're on to our last step. Woot woot. So this one, we're going to use acyl-CoA acetyltransferase. Keep in mind that this is a thiolase. Thio is sulfur. You know how Cohen likes sulfur. Lase means to cut. So you're going to use something to cut our previous product. We're going to, with the addition of CoAsh, our end product is going to, there's actually going to be 2 products. We started off with a C16 fatty acyl-CoA. Now we've removed 2 carbons, so now we're going to have a C14 fatty acyl-CoA. more importantly, we're going to get our acetyl-CoA. And I'm just going to draw out the structure because I want to show you what happened. But this is acetyl-CoA. So from our previous step, we can see that it obviously split. Our end product, you can basically think, is this of our original C16 fatty acyl-CoA. This came in, donated its hydrogen, and attached to the new C14, and that's what gave us the acetyl-CoA and our new fatty acyl-CoA. In this step, know that thiolysis happened. You use the sulfur to cut it. So you'd have a fatty acyl-CoA and your acetyl-CoA. This will repeat many, many times until all you'd have left are acetyl-CoAs. But this is only good if it's an even number of fatty acids. So C16 would produce 8 acetyl-CoA. But if you had C17, well, you'd go through beta-oxidation like we just did. So and depending on how long your fatty acid was, you're going to end up getting N, the number of acetyl-CoAs. So N acetyl-CoAs. But you're also going to get a propanol-CoA, which is a 3 carbon long. So I'm going to put I think that's how you spell it. So this is 3 carbons long. Just keep that in mind. So you're going to have a long odd chain fatty acyl-CoA. It's going to go through regular beta oxidation, end up with N-acetyl-CoAs, but then it's going to end up with propanol-CoA, which is 3 carbons long. Then you're going to use propanol-CoA decarboxylase. Carboxylase, excuse me. And I can't spell right now. And when you use carboxylase, you're going to add in CO2 and ATP. And this is going to give methylmalonyl-CoA. And this may not mean anything to you, but in odd fatty acyl-CoA beta oxidation, you must keep in mind the most important thing is that you use B12 and biotin. Star it, love it. Know that this happens in odd fatty acyl-CoA beta oxidation. So with the B12 and biotin, you'll take this and it'll turn into succinyl-CoA. And succinyl-CoA and our acetyl-CoA that we got from our even and odd beta oxidation, they can both go into the Krebs cycle. And this is great because now we can get the energy from the main source of triglycerides, the fatty acids. So with through all these processes that we just did with the glycerol metabolism, fatty acid activation, transfer of fatty acids into the mitochondria, and beta-oxidation, we've ended up with molecules that can either be in glycolysis. With glycerol metabolism, we ended up going through glycolysis. And now with beta-oxidation, we can go through the Krebs cycle, TCA, citric acid cycle, however you want to say it. Hopefully by the end of this, you have a great flow chart so you can see how all of these are connected. If not, in just a second, I'll show you what mine looks like, and you can study off it. Good luck on your exam tomorrow, guys. I really hope that you all do awesome. Keep in mind, though, that some of these products can go into ketogenesis, and Dane went over that, so study hard, guys. Good luck. Hey guys, it's me, Chris again. I just wanted to show you my chart that I used to study off of for Cohen's part of the exam for triglyceride catabolism. So basically I went through his notes and I wrote down things I thought would be important, like the major source of energies from fatty acids, and the liver and heart and resting skeletal muscle use the fatty acid as a dietary source. Then I also went through the flow chart. So here I'm going to zoom out. This is what it looks like. And now I'm going to zoom back in. Okay, so basically I have triglycerides are broken down to glycerol and fatty acids with hormone-sensitive lipase. Over here I have my glycerol metabolism, and over here I have fatty acid activation, the transport of mitochondria into the transport of fatty acids into the mitochondria, and now I have beta oxidation, and then beta oxidation with odd chain fatty acids. And I also have where they lead to. They can either lead into the TCA, Krebs cycle, citric acid cycle, and that word that you can't read is ketogenesis. So I hope you guys made a chart that looks similar to this. Good luck, guys. We all want you to do well.